quite. So today we're going to start a new section of the course, the final section of the course on the digestive system, trying to learn more about how we get nutrients from food. Before I go on, I want to make an announcement. I'm not sure if this is a reminder or a first announcement, but today at noon in CST 300, Dr. Abraham Polanco Safedit, Pavel's brother, is going to be presenting a talk on the path to becoming a cosmetic surgeon and how to use skills to strengthen the community. And he's going to be giving his talk both in English and in Spanish. So learn or practice your professional Spanish. There will be an opportunity to do that. And it's being sponsored by the college, by the Center for Global Health, by Amigos, by Wayne County. So I think it'll be, it's obviously not just for our own students. This is a community presentation. Anyway, that's noon today, CST 300.
Did you say that he was going to be in our lab?
Pardon me?
Did you say he was going to come to our lab?
There's Peter or Mike, one of them, mentioned to me that he was interested in dropping in on the cadaver lab. Whether he will have time with all of these sponsors who want a piece of him to do that is unclear. So he hasn't scheduled a drop-in time, but he has expressed an interest, and we'll see. Okay, so today, like I said, we're transitioning to the digestive system, the gastrointestinal system for digestion of food. And so today what we're going to do is get an overview and talk about, we can do this by focusing on food groups and the digestion of the various food groups, or we can do this by focusing on the organs of the digestive system and how they digest, what they do, their anatomy and physiology to digest food. And I'm going to do this both ways. Your book does it, it starts with a digestion of food and then it goes back, sort of circles back and does it with a digestion, from an anatomical and physiological perspective. And I guess I'm kind of doing the same thing. So we'll talk today, we'll get an overview that will then start to expand from the anatomical and physiological perspective. So we'll talk about the digestion of carbohydrates, proteins, and fat. There are three major food groups that we eat. We'll talk some cellular anatomy, a layered cellular structure that is common throughout the digestive tract. And we'll start to talk about some exocrine secretions and their functions. Specifically, we'll talk about saliva and its exocrine secretion and its role, and then deglutition, which is a fancy word for swallowing. Okay, so you already know a lot about the digestive tract. You've seen it in the lab. At least you've all seen some of it, and some of you have seen almost all of it, and we'll be doing some more of that today, this afternoon. By the way, I sent out an email earlier this week giving you assigned 1-hour slots the same way we did last week. It was a new randomized list, so some of you are in a different slot, but some of you are in the same slot you were in last week. If you have any questions about when you should come to lab, just ask me. So here's an ad that I scanned a few years ago. This is for a drug that acts to help lower serum cholesterol because high cholesterol is associated with heart disease. And so the important thing is not the drug. The important thing was this ad that had a cartoon of the digestive tract, which I thought was particularly bad. I wouldn't expect an anatomically correct digestive tract in an ad. It's a schematic, right? I understand that, but this one seems really, really bad. So let's go around. So there are lots of things missing. So let's just go around the room, because that will help us sort of articulate, what we already know. I didn't bring the laser to hand, so I'm just going to rely on people to volunteer things that are wrong or missing from this picture. Now,
Stomach.
There is no stomach, right? Yeah, no stomach.
If we can distinguish large intestine and small
Yeah, large intestine and small intestine are not really clearly distinguished. That's another wrong thing. There are some, that we can unpack each of these wrong things a little bit more, but that is certainly wrong. Morgan?
It kind of just looks like everything goes straight through. There's no like muscles or any other.
Okay, there's no muscles or anything. There's, I mean, that's true. So we talked about the stomach being missing. Muscles being missing is also true. What else is missing? Me?
Does the digestive tract start with like esophagus?
Okay, so the digestive tract doesn't start here. That's for sure, right?
So there should be a large intestine and a small intestine.
Large intestine and the small intestine. Which one? So this one starts with something large and then gets smaller. That's not the way it works, right? We start with the small intestine and then go to the large intestine. So that's bad.
Also, it seems like the liver is acting as like an actual step in this process.
That's right. So it looks like the liver is part of the main flow of food when it isn't. We'll see that. It's a secretory organ that is important for the digestion of food, but the food doesn't go through the liver on its way through the digestive tract. What else is wrong about the liver? Way too small. Okay. We said that if the digestive tract doesn't start here, what else is wrong about the start and the finish? Well, so it doesn't just end here, right? There's another ending that I understand that they wouldn't cartoon, but let's be clear about that. It doesn't just stop in the middle of the body. What else?
The length of the intestines itself is
Way too short. Pancreas is missing. Completely missing. Sphincters, you mean the sphincters, is that what you said? Oh, the wrinkles, it's more folded than this. Yeah, that's certainly true. I mean, this is trying to cartoon the idea that this is a folded structure. I understand that, but they kind of this wasn't meant for us. This was meant for the general public to try to sell a drug. And so you can sort of see what they did, but I just thought it was an interesting cartoon that was way wrong. So now let's go to something more. It's a cartoon, but it's more anatomically correct. And we're going to see how the mouth and the salivary glands play a role in digestion. We will talk about swallowing, a reflex. Well, part of it is reflex, and part of it is learned. We'll talk about that. That is to say, part of it you were born knowing how to do, and part of it you learned how to do as an infant. Then we'll talk about peristalsis, the muscular contractions necessary to propel food through the esophagus and into the stomach, and then peristalsis will continue throughout the digestive tract to move the digested food, which we will call chyme from now on. Chyme is the word for what the food becomes on its way through the digestive tract. So we'll talk about peristalsis of chyme. We will talk about the main flow from mouth to esophagus, to stomach, to small intestine, to large intestine, and then to the rectum. We will talk about the auxiliary organs, the liver, the gallbladder, and the pancreas that provide excretions that are necessary for the digestion of food on its way through the main pathway. And we'll talk about some endocrinology. We can't do this without talking about some hormones that are necessary for control of these functions. So let's start with food. Let's just start with what we do with food on its way through our digestive tract. So let's break it down. First, let's talk about carbohydrate because it's pretty simple. So there are enzymes, but the most important enzyme for the digestion of starch, carbohydrate, right? Polysaccharides, a baked potato, a piece of bread. The most important enzyme is called amylase. And your saliva contains amylase, and your pancreatic juice that is secreted has amylase in it too. So we talk about salivary and pancreatic amylase. And it cuts starches into disaccharides. It starts in the mouth, continues in the stomach, and then finishes up in the small intestine. Cutting starch, complex carbohydrate, polysaccharides, into 2 sugar units disaccharides. Then there are other enzymes in the small intestine that cut the disaccharides into monosaccharides, so simple sugars, glucose, fructose, galactose are the 3 main simple sugars. The disaccharides that we'll talk about are maltose, sucrose, and lactose. Other enzymes, including lactase, that cut the disaccharides into monosaccharides, and then a sodium-coupled co-transporter mechanism for the absorption of monosaccharides into the blood in the small intestine. So we'll talk about the cellular and molecular details of how monosaccharides get from the lumen of the intestine into the blood. You know, that's where we want it, to go to the liver or to the rest of the body, the brain. Okay, so this one's pretty simple. We will also talk about, and the main organs we're going to talk about here are the mouth, the stomach, and the small intestine. Just to tell you, when we get to those areas, you should be thinking, in part, about the digestion of carbohydrate. Protein is that we start the digestion, well, chewing, chewing a piece of meat or even a piece of tofu is part of the digestive process of digesting protein, mushing it up, mixing it with saliva. But saliva doesn't contain any enzymes specifically geared towards the digestion of protein. The digestion of protein really starts in the stomach with another enzyme called pepsin. And we'll talk about how pepsin is produced from its precursor. It's got a precursor enzyme. And we will talk, so the stomach releases a precursor to pepsin, which is then turned into pepsin, which hydrolyzes proteins, peptides. And we will talk about pancreatic enzymes, enzymes from pancreatic juice, including these names, we'll get to know these names, that digest the protein in the small intestine. So we have pepsin working in the stomach, and we have these enzymes, trypsin, chymotrypsin, carboxypeptidase, and endopeptidases, working to hydrolyze polypeptides, and then oligopeptides, and then dipeptides into amino acids. And then we'll talk again about a sodium-coupled co-transporter mechanism for absorption of amino acids in the small intestine. Okay, so these are pretty straightforward. Fat is more complicated. Fat is not really very water soluble. You know that if you take a stick of butter, well, let's just take a teaspoon of butter, and you mush it up in some water, it just stays, there's butter and there's water. It doesn't dissolve in water very well. So we have a detergent that helps dissolve the fat. Now, fat has a bad reputation of being, bad for you, but it's a really good source of calories, and so we evolved mechanisms for both seeking out fat. we like it. It tastes good. And so not only do we seek it out because it's a rich source of energy, but our body has a very highly adapted mechanism for absorbing it. So the first part is that bile from the liver and gallbladder are going to make an emulsion of fat. So emulsification. The best example of emulsification is mayonnaise. If you take oil and water, or oil and vinegar, and you shake it up, it separates out into oil and vinegar, right? That's, you know, oil and vinegar salad dressing. You mix it up, and then you watch it, and it goes into oil and vinegar at the bottom. If you add egg yolk to oil and vinegar, this is now the basic recipe for mayonnaise, and you know that mayonnaise doesn't separate because the egg yolk helps the fat form an emulsion, sort of a gel of tiny droplets of fat mixed with water, or in this case, you know, vinegar, and in this case it's not an enzyme that is emulsifying the fat. It's just another molecule in egg yolk, an egg white. So egg is an emulsifier of fat and water, and that's what mayonnaise is. So what we do is we secrete bile that's going to mix with the fat, not in the stomach, but in the small intestine, and emulsify it. Turn those blobs of fat from, you know, if you ate a piece of pizza, you know, at lunch today, all that cheese and that oil is fat. I mean, cheese has got a lot of fat in it, and it will form these blobs in your stomach, but then those blobs will turn into small emulsion particles in the small intestine because it's being emulsified by bile. Bile is acting like a detergent, so you may, you know, if you wash dishes and you've got a bowl that had a lot of oil in it, and you try to wash it with just water, you know, it doesn't clean. But detergent will emulsify the fat on the bowl and then solubilize it so that you can rinse it off. So that's what the detergent is doing. So detergents are going to turn these blobs of fat into small emulsion particles, microscopic emulsion particles, and then we're going to have enzymes that are going to work on those because we now have a large surface area that these enzymes can work on. So we're going to talk about lipase from the pancreas. That's going to hydrolyze the fats to fatty acids and monoglycerides. Those small bits, fatty acids are, you know, small bits of fat, and monoglycerides, small bits of fat from the hydrolysis of fat, are going to get absorbed into the intestinal cells of into the epithelial cells of the small intestine by simple diffusion. Sort of, you know, the way carbon dioxide can go wherever it wants, these fats, these small fatty acids can diffuse. Then, now it gets complicated, then inside the intestinal cells, these bits that have been passively absorbed get assembled into a macromolecular structure called chylomicrons. So the cells actually assemble the bits of fat, along with triglycerides, into chylomicrons, a structure, that are then exocytosed from the epithelial cells into the lymphatic system, not directly into blood. So now we're going to introduce a whole new circulatory system, and I'm not going to do it justice, but you've heard of lymph. And now we're going to need to know about lymph, because that's where the fat goes first. And then the lymphatic system is going to drain into a vein, which now is bringing the fat to the circulatory system, where it goes throughout the entire body. So those are the overviews of our digestion of the three main food groups. Now we're going to start to take it apart, you know, at a more molecular, well, anatomical, physiological, and cell and molecular basis, you know, sort of step by step. Any questions about these the way we're approaching this with regard to food. Okay, so here's a figure from your book, which is too small to see. So I've broken it into halves, and I gave you relatively large-scale printouts of it. So this is sort of, we're still in the overview. We're going to start in the mouth, and we're going to talk about the salivary glands. Then we're going to move down the esophagus and talk about mucus again, and peristalsis. Then we're going to go into the stomach, and we're going to talk about gastric acid, pepsin, well, specifically gastric acid and pepsin, focusing on the digestion of protein, mixing it up, the stomach churning, so peristaltic action in the stomach to churn the food into chyme, mixing it with acid and with enzymes to help hydrolyze the protein, but not yet enzymes for fat. Well, a little, so the amylase from the saliva is working in the stomach, but the stomach acid neutralizes it, so it works for a while and then it stops working because of the acid environment. The food is working its way through the stomach into the small intestine. The first section of the small intestine is called the duodenum. These are names, and they're on, so the key concept list and the coloring book will be your guide to what you need to know for the anatomy section of this, the anatomy part of this section of the course. The pancreas, so the food does not actually go through the pancreas. The pancreas is a secretory organ. It secretes, and we're going to focus mostly on its exocrine secretions, the secretion of digestive juices from the pancreas that then go into this duct, the pancreatic duct, and empty into the small intestine. And those pancreatic juices mix with the chyme that comes from the stomach to continue the digestion of the chyme in the small intestine. So we'll talk about exocrine secretions in the pancreas. You may also know that the pancreas is the source of endocrine secretions of insulin and glucagon, and we'll talk about that at the end of the section. Remember, what's the difference between an exocrine secretion and an endocrine secretion? Let's get that out on the table. Anybody want to try to answer that one? Into what do we secrete hormones in an endocrine secretion? Into blood. Hormones are secreted into blood. Into what do we secrete an exocrine secretion?
Outside of the blood?
Outside of the blood, yeah, but we can be more, that's correct, but that's not complete. Where, it's not in the blood, okay, so that, it's not blood. Where do we secrete the exocrine secretions? Outside, into the digestive tract itself, into ducts that then drain this fluid. So the salivary glands are exocrine organs that secrete saliva into the mouth. The pancreas is an exocrine organ that secretes pancreatic juice into the small intestine. Okay, so exocrine, sweat is an exocrine organ. I mean, the skin exocrine functions. The sweat glands secrete sweat, fluid, salt, water, onto the surface of the skin to help cool the body. So sweat glands are an exocrine organ. Anything that is secreting something into a duct that then goes somewhere else is exocrine. So we're going to be talking about acid, and then we're going to be talking about so the pancreatic juice is going to contain enzymes. Let's see, can I remember some of them? Amylase, the precursors to trypsin, chymotrypsin, and carboxypeptidase. So the enzymes necessary for carbohydrate and protein digestion, and lipase, necessary for fat digestion. So all of that is present in the enzyme mix that the pancreas secretes. And it also secretes bicarbonate, which is going to be important for neutralizing the acid. The stomach is specialized for an acidic environment, but the intestines are not. If gastric acid goes through the intestines for too long, it actually causes an ulcer, and we have a mechanism for neutralizing the stomach acid with bicarbonate. Remember, bicarbonate is a weak base, so mixing a strong acid with a weak base, we're gonna neutralize it. Okay, continuing down, so here, this is the small intestine, and we'll talk about its functions in detail. Here is the liver. The liver is, for most of what we're going to be talking about, well, we'll be talking about both the endocrine and the exocrine functions of the liver. The liver is going to produce the bile that is going to be stored in the gallbladder and then secreted, or exocrine secreted into the, this is where the figure gets sort of cuts badly. The liver, there's the tube from the liver, is also going to secrete exocrine juices into bile from the liver and gallbladder into the small intestine. And we'll see a whole bunch, 10 major functions of the liver, both exocrine and endocrine and digestive, just sort of storage. Liver is a place where we store a lot of our food between meals. Then the large intestine, so small intestine first, and we'll talk about the different segments, duodenum, jejunum, ileum, and then the large intestine. We'll talk about the specific parts of the large intestine, which are mostly to reabsorb the water that was secreted, the large amount of water that was secreted each at a time during the upstage, during the processes in the stomach and in the small intestine. The pancreatic juice secretes a lot of water. We want to recover that water, and that's a function of the large intestine. Okay, so let's talk about this general layered structure of the digestive tract. It's more complicated than any structure we've been talking about so far. And I do want you to know the details on this slide. It's on the key concepts list, and it's on this slide. So we're going to start, so let's get oriented. Up here is the lumen, the inside of the tube of the digestive tract. And we're just talking about, this is generic. This could be stomach, this could be small intestine. Could be large intestine. We'll talk about specifics when we get to each of those sections. This is generic. So we've got a layer of epithelial cells that line the lumen of the small intestine, or the stomach, or the large intestine. Epithelial. You already know about epithelial cells from the kidney, but these are specialized not for reabsorption of things like in the kidney, but for absorption of food groups. The structure is going to be highly folded in order to increase the surface area. And then those folds will have microscopic folds. And then each cell will have its own microscopic folds called microvilli in order to increase the surface area. But we will talk about folds upwards and then sort of inward folds that are sometimes in some places called crypts. So we're going to talk about villi, which is the plural of villus, and crypts. Villi are the outward folds, and crypts are the sort of the inward folds.
Are they both microscopic? Or?
No, some of them. So we're going to see big ones that are visible. Well, actually, the villi and the crypts are microscopic. The folds are macroscopic, and we'll see those today. Mixed in with the epithelial cells are endocrine and exocrine cells. Remember, endocrine cells secrete into blood, and exocrine cells secrete into the lumen. We're also going to see cells that secrete mucus, just like and they're called goblet cells, just like the cells of the respiratory tract. We're going to see ducts from the exocrine organs, like from the pancreas, or from the gallbladder, or from the liver, or from the salivary glands, in specific areas. So, and these duct cells are not just plumbing, they're not just a tube, they also do their own secretions. So we'll see, for example, that pancreatic duct cells are important for secretion of some of the things that the pancreas secretes, and then the other parts of the pancreas secrete the other things that the pancreas secretes. So the duct cells are not just providing a tube, but are part of the secretory process. Underneath the epithelial layer, which also has the endocrine and exocrine cells, is a layer called the lamina propria. So the lamina propria is the equivalent of the interstitial space that we've been talking about in the kidney. It's not cellular, it's just connective tissue between the epithelial layer and then the other layers. So this, just like in the kidney pictures, this lamina propria layer is exaggerated in its thickness. It's an interstitial layer that in this cartoon is exaggerated in size. Underneath the lamina propria is a layer of muscle called the muscularis mucosa. This whole, muscle is going to be important for peristalsis. So this is the first layer of muscle that is important for peristalsis. So I'm using some words here. Peristalsis. Does everybody know what I mean when I say peristalsis? It's the sequential squeezing of a tube. So when you squeeze a tube of toothpaste from the bottom, as you know, over the course of the months that you're using a tube of toothpaste, and you're squeezing it towards the nozzle, you're doing peristalsis. You're squeezing it from the bottom and then slowly squeezing it towards the top. That's a peristaltic action. And that's what the digestive tract is really good at. The sequenced, coordinated squeezing pushes the contents forward and prevents it from being pushed backwards. If you squeeze a tube of toothpaste in the middle, the toothpaste goes in both directions. But if you squeeze it carefully from the end, you can get your toothpaste out efficiently out the nozzle. Same idea here. I should have brought a tube of toothpaste in to demonstrate.
How do you spell
peristalsis? And so we'll talk about peristaltic movement due to peristalsis. So the muscularis mucosa is part of the muscle system that does peristalsis. This whole area, the lamina propria and the muscularis mucosa, is called the mucosa, the mucosal layer. Underneath the mucosal layer is the submucosal layer, which that's a helpful name, submucosa, and that consists of circulatory system and lymphatic system microvessels. So we're talking about capillaries here. Capillaries and microscopic lymphatic vessels. Really tiny ones. And underneath these circulatory system vessels is the submucosal nerve plexus. So these symbols here are supposed to indicate neurons. This is a neuronal cell body, here's a neuronal axon, here's a neuronal presynaptic terminal, and then here's the cell body and dendrites of the next neuron in this network. That's what plexus means, network of neurons. So the nervous system both the submucosal nerve plexus and the myenteric nerve plexus is an important part of controlling the muscles so that they contract when they're supposed to in order to do efficient peristalsis. So it turns out you have, a lot of neurons in your digestive tract. And it's a whole nother nervous system. So we talk about the central nervous system and the peripheral nervous system, but now we have a digestive nervous system controlling the musculature that controls peristalsis in the digestive tract. But these two parts form the submucosa, and then under the submucosa is another layer of muscle and another layer of muscle and another nerve plexus for doing more control of contraction for peristalsis. So we got this really highly developed system for moving chyme through the digestive tract. And then out here is a layer called the serosa. So this is anatomy. This is cellular anatomy of the digestive tract. And your coloring book has a figure that is part of the assignment, and this slide will be helpful to you. And the key concepts list has a list of these things too. So understanding the organized, layered structure of the digestive tract. And we'll see specifics of this, like when we're in the large intestine, we'll see specific folds, and villi and crypts and stomach. The stomach is a little different from the small intestine, and the small intestine is a little different from the large intestine. But these are the generic structures. Okay. So let's talk about saliva. So now we're going to start our trip through the digestive tract. There's actually a pretty good book. It's called Gulp, G-U-L-P, and it's written by the same author, whose name is escaping me right now, who wrote another book called Stiff, which is about cadavers. And she's a really humorous writer. So she writes about, Gulp is a trip down the alimentary canal, the digestive tract. And it's pretty entertaining. So if you want some entertaining Thanksgiving reading while you're digesting your Thanksgiving food, go to the library or go to, get Gulp. It's entertaining. And it's actually factually informative. So this author does a good job of combining science, knowledge, in a very entertaining entertaining context. Okay, so here is a figure of the mouth. We have 3 salivary glands, names that I do want you to know. The parotid gland, parotid, around the ear, so back here. The parotid gland, the submandibular gland under the mandible, so under the jaw, submandibular, and sublingual, under the tongue. 3 glands that secrete saliva. And you know what saliva does. It lubricates, when you're chewing food, you mix the food with saliva to help it, get lubricated and get less solid and more, doesn't become liquid, but it becomes lubricated, mixed together, more of a gel than a solid. Think about if you eat something solid, let's say a piece of meat. That piece of meat is pretty solid, but you chew it and it becomes less solid. You all know what I'm talking about. So the saliva is used during chewing to mix the food into a more swallowable bit. It initiates the digestion of carbohydrates because of amylase, and lipids and fats because of lipase. So it's not just secreting water, it's secreting some enzymes. And it also has some other functions that we're not going to focus on very much, like it does secrete some immunoglobulin, so it acts as a sort of a protective secretion. animals use this. You know, you've seen dogs, you know, if they get a wound, dogs will lick their wound. That's not just cleaning it, it's also trying to fix it with protective saliva. And if you eat something acidic, or you eat some, which is not a problem because it's going to the stomach, which is acidic, but if you eat something basic, it helps neutralize those acid and basic foods so that they don't cause problems on their way down the esophagus. Okay, that's saliva. So now we're focusing in on the salivary glands. And so we don't need to know great detail about the salivary glands. So nothing on this slide is required names, except for the fact that the duct cells are epithelial cells that do stuff. And then the dead end cells at the ends of the ducts, which have a name, they're called acinar cells, also secrete stuff. So, and they are also lined with epithelial cells. So you can see here is a cross-section through a duct, and here is the apical membrane and the basolateral membrane of this epithelium that surrounds the duct. And here is a cross-section through a part of the acinus with, again, an apical and a basolateral membrane of the epithelium. So in the kidney, the epitheliums are specialized for reabsorption of materials. But in these glands, the epithelium are specialized for secretion of material into the ducts necessary for digestion. And we'll get into some of those cellular and molecular details, which you all have learned to love after break. So in the saliva, so we have 3 salivary glands. They are all slightly different. Saliva has both a watery content and a mucus content. I guess you do not have to know the proportions of the different types of cells that secrete a watery saliva or a mucousy saliva. But I guess I do want you to know that there is a watery saliva secreted by the serous cells and a mucousy saliva secreted by the mucus cells of the salivary glands. And it just so happens that the sublingual salivary gland secretes a lot of mucousy saliva, and the parotid gland secretes a lot of watery saliva. So let's watch a video about deglutition. Swallow.
Swallowing, or deglutition, is the process by which food passes from the mouth to the stomach. This movement is controlled by the nervous system and involves both voluntary and involuntary muscle contractions. Before food is swallowed, it is mechanically broken down in the mouth by chewing or mastication. The salivary glands secrete the enzymes lingual lipase and salivary amylase. Salivary amylase begins the chemical breakdown of starch, carbohydrates, in the mouth. The water content of saliva helps bind particles together to form a bolus that can be swallowed. Swallowing occurs in 3 stages. The first is a voluntary phase called the buccal or oral transit phase. Here the tongue moves upwards and backwards against the hard and soft palate. to push the bolus into the oropharynx. Next is the pharyngeal stage, in which involuntary movements push the bolus through the pharynx and into the esophagus. Movement of the bolus stimulates receptors in the oropharynx, which send impulses to the deglutition center in the medulla oblongata and the lower pons of the brainstem. The brain signals the soft palate and uvula to close off the nasopharynx, and the epiglottis to seal off the larynx. This prevents the bolus from entering the respiratory tract. The upper esophageal sphincter relaxes to allow the bolus to move into the esophagus and contracts to reduce backflow into the pharynx. In the final esophageal stage, the bolus is pushed onward by peristalsis, a progression of involuntary wave-like contractions of the circular and longitudinal smooth muscles of the esophagus. As the bolus approaches the end of the esophagus, the lower esophageal sphincter relaxes and the bolus moves into the stomach. So, in summary, food moves from the mouth through the oropharynx and esophagus and then into the stomach.
Okay, that's enough. Do any more? So that, the URL for that video is on the, this is an interesting set of videos that I've watched, I guess. So that video describes swallowing pretty well. You know that some of it is involuntary and innate. Newborns know how to swallow liquid, right? Newborns do not know how to chew food. How many of you have ever watched a baby try to process their first bites of solid food, baby food? What did you see?
Just like kind of mashing on it, like didn't really know how to chew.
They didn't know how to chew.
It just kind of.
It
Wasn't
goes
sure.
everywhere. It's
Yeah.
all over their face because they don't know how to move it in their mouth to the back of their mouth. They, you know, it's a learned process of being able to form a well-formed bolus, push it to the back of the throat, and then swallow it. We aren't born knowing how to do that. We are born knowing how to get to swallow a liquid because we don't form a bolus of liquid, we just swallow. So some of it is innate, and some of it is learned, especially chewing is learned. There are some, hilarious videos of, babies trying their first, baby food of peas, and just, it's everywhere. And then eventually it's not everywhere. Eventually they learn, okay, I got this thing in my mouth, do these movements, and I can get it down into my stomach where I want it. Well, hopefully that's where they go. Okay, so let's take a break. And the rest of this class is just a series of clickers, not so much to check your knowledge, but to use as a basis of sort of a new basis for learning. Okay, so let's take a 5-minute break, and then we'll do a series of clickers. Let's do a bunch of clickers. Some of these are review, and some of these are not review. I'm asking you to figure stuff out. So this is a review. Which of the following is true about amylase? Okay, seeing some correct answers. A and B are both correct, but C and D are both false. Amylase is important for the digestion of carbohydrates, not proteins or fat. And it is secreted, there's salivary amylase and pancreatic amylase. So, salivary glands and pancreas. Okay, which of the following is true about pancreatic secretions? And this one, they're all true. Pancreas secretes bicarbonate, which neutralizes gastric acid from the stomach. It secretes lipase, which is important for the digestion of fats. It secretes precursors to enzymes that digest peptides, polypeptides, into dipeptides and amino acids. So those are the enzymes like trypsin and chymotrypsin and carboxypeptidase. We'll talk about more of those later. We'll talk more about those later. And digestion of carbohydrate, because it secretes amylase. So all of these are true about the pancreatic juice. The main function of gastric acid is, so this is the acid, stomach acid. And even though it suggests that there's one correct answer, there are two. There are two main functions of gastric acid. One of which I told you about and one of which you may not have heard about yet. So this is not a completely review question. So this is a question to see how much some of you already know walking into the room from high school or from other classes. So what's the one that you're all pretty confident of? Oh, protein, hydrolyzed protein, and it activates pepsin. So the acid is necessary to activate an enzyme that is used for the digestion of protein. Carbs are pretty resistant to acid hydrolysis. And the acid then neutralizes the amylase. I mean, amylase doesn't work at the pH 2 that our stomachs are at very well. So the acid is not really specialized for digestion of carbohydrate, although there is some acid hydrolysis of carbohydrate too. Okay, the main function of bile, which is made by the liver, stored in the gallbladder, and then secreted as needed during a meal, during digestion of a meal by the gallbladder. And we'll talk more about that after break. Solubilized fat. Bile is for fat digestion. Okay, so now we're getting into some things that I didn't teach you, and these clickers are meant to remind me to teach you, and to also just see if you can, what your gut reaction is to these questions. What volume of saliva is secreted per day in a normal adult human being? Show me what I didn't teach you. You may not know. I'm just curious what you think. What's your estimate based on what you know? Lots of you are getting it right. 1 to 2 liters. Now you know what a liter looks like. Who's got a water bottle? There's a liter of water, right? Morgan's got a liter of water. 1 to 2 of those of spit every day. Amazing, isn't it? Okay, here's another one that I didn't teach you, but this is to get this conversation going. what stimulates the secretion of saliva? So some of you who have some neurophysiology will know about this. Best answer is B, the parasympathetic nervous system. Remember, the sympathetic nervous system is the fight and flight nervous system, and the parasympathetic nervous system is the rest and digest nervous system. So here we are in digestion, so we have to be thinking about the parasympathetic nervous system. This is the so-called cephalic phase of digestion. Cephalic. Head, brain, the brain phase of digestion. I can get you to start secreting saliva right now using your brain. I can start describing the smell of pizza that is starting to waft down the hallway at lunch. I can remind you of the smell of turkey cooking in your mother's kitchen that you'll be eating next week. Lasagna, just lasagna, just the smell of lasagna. I'm doing it. I can feel my mouth watering because I'm talking about food. And I bet that is happening to some of you as well. Watering, mouth watering is stimulation of saliva. That's your nervous system, especially your parasympathetic nervous system, activating the secretion of saliva. The acetylcholine is binding to a type of GPCR that is coupled to GQ. Calcium exocytosis of salivary products. The sympathetic nervous system is more complicated. The sympathetic nervous system activates the secretion of mucus saliva, and it inhibits the secretion of serous, watery saliva. So some of you may know that when you're highly stressed, your mouth gets sort of sticky and dry. That's the sympathetic nervous system drying out your mouth with a mucousy saliva rather than a serous, watery saliva. And these are hormones that we will be talking about. Well, this one we've already talked about. You know what that does. And cholecystokinin is a hormone that is going to be important in the digestive tract, but not for the secretion of saliva. Okay, so some new concepts here. The brain affecting digestion. Oh, that's the last one. So we're done early today. That's convenient. I'll see you in lab this afternoon. Remember Dr. Abraham's presentation at noon. I'm happy in lab today we'll be exploring, we'll be opening up the digestive tract more and more, and by the end of the afternoon today, I think we will have the digestive tract sort of fully open. Then the week after break, we will circle back and we will use that opportunity with the cadaver to ask and answer questions related to the practical quiz that is coming up the week after that.